What does the parasite do to make the red cell feel like home?
Puts in a couple of love seeds, a nice rug.
Hello, everyone, and welcome back to People, Parasites, and Plagues, a podcast aimed at delivering information about the fascinating pathogens among us from the scientists who study them. I am David Peterson.
And I am Kim Klonowski, your host for today's episode. So what happens when a parasite messes with the wrong college student? In today's episode, we'll find out.
Our guest today is Dr. Vasant Muralidharan, Associate Professor in the Department of Cellular Biology and a member of the Center for Tropical and Emerging Global Diseases. Vasant, welcome to the podcast.
It's great to be here. Thanks for inviting me.
So Vasant, one of the first things we ask our guests about is their pathway to science. Can you talk a little bit about what your journey looked like?
Okay, so my journey was not straight. It was a little meandering. I will say that I was not a kid who was too interested in science growing up. I was more interested in being outside, playing sports, or roaming the hills near where I grew up. And my interest really grew from this fascination with nature. So I was always interested in wildlife and animals, and I would roam around counting the birds I saw and the types of birds I saw. And after finishing high school in India, where I grew up, your choices usually are restricted to either medicine or engineering. And like a good kid, I started in engineering college first. And because I knew I didn't want to do medicine. My older brother is an MD, and I had spent a summer with him while I was in high school while he was doing his internship, and very quickly realized that I would be no good with patients because I don't have a lot of patience. And so I knew medicine was not for me. And so I said, well, what else was left? And engineering was an option that was offered, so I took it. But 6 months in, I realized engineering is also not for me. I absolutely hated it. And I dropped out of college, which was not something that was done at the time. I guess they call it a gap year now. But at the time, nobody was happy with me, including my parents, who thought I was wasting my life, which I probably was at that time. And then I restarted because my dad basically told me, look, I need you to finish college, do something. So just go join a Bachelor of Science program or something and just do that. And so I did that. And luckily for me, I found mentors there, especially one I would like to mention, Dr. Nair. She just recently retired, and I was lucky enough to be there for her retirement party. She saw something in me that I hadn't seen in myself, which was this curiosity and the fact that apparently I was smart, but I did not have a good work ethic, is what I would say at the time, right? And so she really pushed me to develop that work ethic that is needed to go along with your sort of, curiosity and intelligence. And that really pushed me hard to dig deeper into what I was interested in, what sort of science I was interested in. And this was, I was majoring in chemistry and microbiology at the time. And I was really fascinated by the world of proteins and how they function. So that's where I decided, okay, I'm going to do science. I didn't know what my path would be or where I would go, but I knew I was interested in it. And this was something that was really driving me to do better myself in ways that I hadn't anticipated. And so I said, well, let's see how far this takes me. And apparently it's taken me to be an associate professor in the Department of Cellular Biology at the University of Georgia, and hopefully professor in the future.
But how did you make the decision to go to graduate school?
So that was an interesting choice as well. So when I was sort of a college student, my professors sort of pushed me to do sort of research in areas I was interested in. And at the time, you have to remember, this was pre sort of online world really exploding, right? So a lot of resources weren't available online. And so to read scientific magazines, I actually had to go to consulates in Bombay. So I went to the US consulate and British consulate, both of which had free copies of Nature or Science that I could check out and read. And so I started doing that, and I was like, okay, this is really interesting. How are these people who are publishing these articles that I'm interested in, how are they figuring these things out, right? How are they figuring what that process looks like? And so then I was connected with somebody who was doing research in India in a place called National Center for Biological Sciences, in a place in the south called Bangalore. And so I went there. I emailed the person, said, can I come do a summer research project with you? And he said, sure, come on over. And then I decided I couldn't wait till summer. So in India, you get like a month and a half long break in winter. And so I literally showed up in his office. Remember, I'm traveling to a different city now, right? So I showed up in his office and I say, hey, remember I'd emailed you about working with you? Can I do it now? And he was very kind and he said, sure, go work with one of my senior graduate students. And I did it. I ran my first gel. I washed my first beakers and I was hooked. I was like, okay, this is what I really want to do. And at that time, I finished my master's, and the same person said, why don't you come to this conference that I'm hosting in India from the Protein Society? And a lot of international people are going to be there, so present your work there, which I did. And I met a couple of professors from the U.S. I did not quite know that there was a path to do a PhD. I thought I'd do a master's and then maybe get a job. But these two, one of them, who's now, I think, a dean at Vanderbilt, he was like, so you seem like a smart kid. What do you want to do after you finish your master's? And I said, I was going to do a job. He said, no, you should do a PhD and you should come to the US to do a PhD. And so I took his advice, applied to a bunch of places, and he came back to India for a different conference and interviewed me for the university I ended up in, which was Rockefeller University in New York. And this interview was basically held in a cab ride because he had to be somewhere and couldn't sit down and interview me. So he took me on a 3-hour cab ride through rush hour traffic in Bombay, which was pretty nerve-wracking because this person at the time was an HHMI investigator, which I think both of you know what that means, right? Mm-hmm. And he was shooting rapid-fire questions at me in this cab, so I couldn't leave. I was trapped. But at the end of about 2 hours, he said, I think you'll do well in the U.S. I think you'll do well with the PhD. You should come to the U.S. And so that was sort of my thought process. So I will say I never had a master plan. I didn't think even at that time, I'm still surprised that I'm a professor teaching college at this time. If you go ask my high school friends what I'm doing right now, nobody probably would have ever predicted any of this, right? So I just said, what do I do next? And I did that, right? So whatever interests me next, that's what I do. And that one step at a time brought me this far. And the one, I would say, arc or a thread that runs through that entire process is I always followed what I was curious about and what I was excited about. And those two things have led me to where I am with the two of you here.
So you may not have had a plan, but it sounds like you had some good mentors along the way.
Yeah, so I had really amazing people who took the time out of their day and sort of took a naive, 19, 20-year-old under their wing and said, okay, you seem enthusiastic. Let's channel your enthusiasm in the right way, right? And I have really appreciated that. I've kept in touch with a lot of them over the years, and I try to pay it forward, right? That is one of the things I've found real joy in. That was, again, not something I expected. This was sort of one of the things I found out as I went through graduate school and postdoctoral research, and now as a faculty, is I really enjoy working with enthusiastic young people and giving them opportunities that other people had given me, right?
So before we get into asking about your research, which is on malaria, maybe you could just give us a quick overview of how big a problem malaria is, and after over a century of research on it, why it's still such a problem.
Let me address the second question first. Why it's such a big problem? I think the key reason there is that malaria has had a head start on us, right? We've only been studying it for about 100 years. But malaria has evolved with humans as a species since before we were a species, right? It has infected apes forever. Before that, old monkeys, it has infected them forever. And so as humans have grown as a species, malaria has been a constant companion for us. It is what I would propose is probably the world's oldest pandemic. We don't think of it as a pandemic because it's not present in great numbers in the US and Western Europe. But for the rest of the tropical and subtropical parts of the world, it is a pandemic. It has existed. It kills more than 600,000 people every single year. It infects 250 million people every single year. And these are numbers that we have reached after a lot of research, after applying a lot of drugs, insecticide-treated bed nets in the field. 15, 20 years ago, these numbers were way higher. It was more like 500 million people every year, and 1 million deaths or more every single year. So it is a really challenging problem for those reasons. The other main problem with malaria is that it's a eukaryotic protozoan. What do I mean by that? It's a eukaryote, so its cells shares a lot of characteristics with our own cells. So to kill something specifically that looks a lot like us is really difficult and challenging, unlike a virus or bacteria, which don't have as many genes as human cells do, right? So that makes it really challenging. And as I said, it has had a head start on us, right? So evolution is a wonderful experiment that we're still learning about, right? And so this experiment of malaria in humans has been going on for eons. And so this co-evolutionary relationship we have with malaria, it has developed a lot of tools to infect us and cause disease. Like most infectious diseases, the goal is eventually to reach an understanding with the host so that they don't actually kill the host and the host can survive to transmit the disease. And I think there are several different species of malaria. The oldest one that infects us is called Plasmodium vivax. It is one of the two major species that cause human malaria. And it has reached that understanding with the human host. Vivax doesn't kill many people. It'll cause a lot of issues for you. It is very debilitating, but it will not kill the host. Falciparum made the jump from apes to humans very recently, only about 1 million or so years ago. So it hasn't had that adjustment period with us so far. So therefore, it kills a lot more people. It kills most of the 600,000 people who die, mostly 2/3 of them kids under the age of 5. They're all infected by Plasmodium falciparum.
So what is the actual fundamental question that drives your research currently?
So our sort of fundamental question that's driving the research that we do in our lab is trying to understand how the parasite interacts with the host cell. What are the molecular methods that the parasite uses to modify the host, to change the host? And when I say host, Plasmodium grows within human red blood cells. That's how it causes disease. And we are trying to understand, to grow within human red blood cells, it needs to change red blood cell physiology completely. And we don't quite understand how it's doing that. Who are the genes in the parasite genome that help it do certain specific types of modifications in the host cell? That's a pathway. If we can block that process, obviously the parasite will lose its ability to grow within human red blood cells. It's not just to grow within human red blood cells. It is to get inside the human red blood cells. It modifies the host as well. It sends several dozens of proteins into the host cell before it actually goes inside the host cell. And we're trying to understand how does it do that? Because proteins cannot just go across biological membranes. They have a specific aqueous channel that needs to be formed to send them through. And we don't know how the parasite's doing that. There are specialized machinery that the parasite has that it uses to modify the host. That machinery still remains fairly undiscovered. And so we are trying to really understand how the parasite's sending these proteins into the host cell, what those proteins in the host cell are actually doing. And remember, at the end of the life cycle in the red blood cell, it actually forms couple of dozen daughter cells that now need to get out of the host cell to go back into a new one.
To continue the infection.
Exactly. And so how are they getting out? Because they need to break down the red blood cell. That process, while in the past decade or so we've identified a few players in that pathway, we still don't know what triggers it. How does the parasite know it's finished cell division? It needs to go out. What's that trigger? What's that signal that it gets that says, all right, we're all done making our babies now. These babies need to get out. And how are they getting out? We don't understand that process very well. And so we take a fairly simple approach to study that. We just look at it using different types of tools and techniques, and just observe how the parasite's doing it. And that has taught us a few things over the years. So that's really driving our interest, is across this life cycle, how is it sensing the host? How is it modifying the host? And how does it know when to do what in the host? That is one of those sort of overarching questions.
So the idea being, if you understand what those things are, those factors are, you can then target those factors in some way.
Exactly. So if we can prevent it from going inside the red blood cell, it's trapped outside. And the reason it goes inside the red blood cell is to hide from the immune system. And so if you can trap it outside, now the immune system has access to all the possible antigens that the parasite has, and hopefully generate a good response so that the next time it sees a parasite, it'll block it in its tracks, right?
Mm-hmm.
So once the parasite gets inside of the red blood cell, it starts to modify it by secreting proteins into the red cell. How do you identify what proteins are secreted into the red cell?
Oh, that's a great question. It is a painstaking process. So 15 years or 20 years ago, roughly, we identified a signal some of these proteins have that go into the red blood cell. That helped us identify a big group of these proteins are sent into the red blood cell. But then it was also discovered that there's a whole bunch of proteins that go to the red blood cell don't have the signal at all. And even the ones that have a signal, some of them actually don't go into the red blood cells. And so it becomes really complicated really fast. How does the parasite know who to send out and who to keep within itself? One of the approaches we took was we said, okay, is there a way we can mark these proteins? Can we put a stamp on them that says, okay, you are exported and you're not exported? And so to do that, we took an approach that uses something called biotinylation, proximity biotinylation, where we put an enzyme on an exported protein that's sent out into the host cell. And then when we add biotin to the parasites, this enzyme will just make a biotin free radical that tags everything nearby. This free radical has a very short time of action, so it basically will only last for whatever is very close to the protein that's being sent out.
So when you say nearby, what scale are we talking about?
Oh, we're talking like maybe 10, 20 nanometers.
Okay. So 10, 20 billionths of a meter.
Exactly. 10 to 20 billionths of a meter. That's a great way to put it. And so now what happens is we put it on one of these exported proteins that we had known from prior research goes out into the red blood cell. And then we tagged it and we basically added biotin when the protein is within the parasite. And then the next time we added it when it was outside the parasite in the red blood cell. And so this way, this would stamp everything that's near it. And then we can use that biotin coupled with mass spectrometry to try and see who has this stamp. And then by comparing the two sort of time points we took, we can say, okay, these proteins are definitely on the outside, right? Mm-hmm. Because we could only see it stamped when our exported protein was on the outside. So this helped identify several proteins that ended up on the outside of the red blood cell. And so now with this work and prior research that's been done in the field, we have a fairly good idea about 400 proteins roughly go out into the host red blood cell. But most of these, we don't know what they're doing actually out there. So that remains the next step to try and figure out what they're doing outside in the red blood cell.
So in the human hosts, the malaria parasite spends its time, as we've discussed, kind of nestled inside this red blood cell. What does the parasite do to make the red cell feel like home?
Puts in a couple of love seeds, a nice rug. I kid, but it sort of does something like that, right? So it installs a new doorway, which allows nutrients to come in that previously couldn't come in, right? So the red blood cell does ion exchange, but it's kind of blind to a lot of big molecules, right? But big molecules need to get in, like cholesterol and other big things, for the parasite to survive. And so the parasite puts in what we call the new permeability pathway. It's just a fancy way of saying I'm adding a door, right? And this door will allow everything that's below like 1,500 Daltons or so to just float right into the parasite. And then it sets up a hub, sort of a protein trafficking hub. can think of it like the Atlanta airport. Things come in, get sorted into different paths, and they go out. And this is kind of a brand new organelle that the parasite sets up. It's called MAR, after an old German scientist who discovered these features in microscopy. And what that does, it modifies the host red blood cell membrane so that the immune system is unable to react fast enough to the parasite. Right? So it also allows the parasite to bind to your cells that line your blood vessels, right? Endothelial cells. So it does that. And it also changes the chemistry of the red blood cells such that a red blood cell, the ion concentrations in the red blood cells are completely flipped compared to a normal red blood cell. And it also sets up a delivery mechanism. So sort of like Uber Eats, right?
These analogies are great.
So what does this Uber Eats actually deliver? It always delivers the exact same thing, which is hemoglobin. And what the parasite does, it literally eats hemoglobin from the inside of the red blood cell. It chews it up in its lysosomes, or we call it the food vacuole or the digestive vacuole. The idea is the same. Hemoglobin from the red blood cell goes into the parasite, into this acidic vacuole where it's chewed up into its bits. And those bits are then used by the parasite to build its own proteins, to make its daughter cells. It needs a lot of resources to build the daughter cells. That's why it's chosen this parasitic lifestyle, right? It's really easy to take somebody else's stuff and build your own stuff, right? And so that's what the parasite's doing. So it's setting up all of these things in the red blood cell. And exactly how it's doing it is a big area of research. We don't have a good accounting or understanding of this.
Wow, this parasite is a real big squatter.
It is, yeah. And it'll rip out all the lead pipes and everything.
And I almost wish this was a YouTube video rather than a podcast, because Vasant has been using his hands to illustrate all of those processes.
Uber Eats, drivers.
And I think it's been very effective.
Very.
I love talking about the parasite. If you don't get that through the sound, I can go on for hours. They'll probably kick me out of this podcast soon.
So one of the challenges of studying the malaria parasite is that they're really small, really, really small. Because we know red cells are small, and these are much smaller than a red cell. And oftentimes in your own work, you're interested in organelles, tinier organelles that are inside this tiny parasite. So what techniques do you do to visualize things inside of the red cell and inside of the parasite?
Yeah, it is sort of a bane of our work life that the parasite's so tiny. It's really hard to see. It is a beautiful parasite, and the way I say that is because we used to do what's called electron microscopy. So this provides the resolution to look at these really small structures. The problem that we frequently used to run into is that electron microscopy, it's very labor-intensive and time-consuming. And you only get a slice of the parasite. So you don't get the full three-dimensional beauty of how things look like on the inside. So it's really hard to say whether, when you do a slice of the parasite, if you're looking for a specific organelle, for instance, did you just miss it in that slice? Or it looks weird because the slice you made had the parasite at a strange angle that you're not used to, right? And so what we and others have started using in the field quite extensively in the more recent years is something called expansion microscopy. So this is a process by which we put our parasites in a little gel, and then we add water to that gel. What that does, it expands the gel. And what's special about this gel is that it expands this with the same exact dimension in every, or same exact factor in every dimension. So if it grows 1 centimeter in the XY dimension, it'll also grow 1 centimeter in the Z dimension. So therefore, this is called isotropic expansion. So therefore, everything expanded the same way in all 3 dimensions. This is important because otherwise you'll get distortions in your parasite if you don't expand in the same direction. It's sort of like stretching an image in weird ways. But however, if you stretch your image in the same way in all directions, then you just expand it. You don't distort it. And so what this has allowed us to do is to use the power of light microscopy, which is not as labor intensive, and we can count lots of cells in light microscopy. And so by doing this, we've been able to observe organelles in detail, in resolutions that was previously not possible. And so this has really been a game changer for us in cell biology, because it now allows you to appreciate structures in ways that wasn't previously not possible, because you can see things that it has upended some of the assumptions we had in the field, which has been really great, right? So every time a new technique comes into a field, it suddenly changes what's possible. It helps you answer questions that you've always had, but never knew how exactly to answer it in a way that was rigorous and reproducible, right? Because that is key in science. Unless your results are the same today versus on Monday versus on Sunday versus on Friday, and they're the same in Athens, Georgia, or New York, or San Francisco, right? It has to be the same exactly everywhere, right? And so that really comes with this ability to get large numbers of data sets, right? So this has really brought that to cell biology, and it has helped us kind of understand things in detail that was previously just not possible.
So with the expansion microscopy, are we talking about 50% bigger, twice as big, or what is it?
Oh, it's about 4 to 5 times as big.
Oh, that's significant.
So now you had a parasite that's basically about a micron size, and now you're expanding it. So what was the red blood cell is about 6 to 7 microns, which is it is probably the smallest cell in the body, and then the parasites within that is about a micron big. Now those parasites are about the size of a red blood cell, which has been really amazing, because now we can see detail, like I said, that we were previously just not able to see. What's more exciting is there are techniques that are coming down the pipeline that'll expand the parasite by about 16 to 20 times.
Wow.
So I cannot wait for that.
That almost sounds like the premise for a horror movie.
Well, these are dead parasites at this time. Once they go into jail, they're dead.
Okay.
So they're not coming for you, David. Don't worry.
Oh my gosh. So entry of the malaria parasite into the red blood cell seems pretty elegant, but egress seems a bit messier. What do we know about this process of egress, and how would you compare it to the entry into the blood cell?
So, yes, you're right. Egress is a destructive process, right? It is the parasite destroys the host cell as it's getting out. And entry is more silent. It's like a cat burglar entering into the apartment. Nobody knows about it. They do their job. But when they leave, it's as if they blew apart the apartment building while they're heading out of the door, right? And so what's common between the two is actually what seems to be certain signaling pathways. So we know they sort of use the same signaling pathways for both these processes. And obviously, invasion follows egress. So what the parasite does is, while it's getting ready for egress, it sets up the merozoid in a way that once it's outside, it doesn't have to do anything other than find a red blood cell and get back in. And it usually does that within a couple of minutes. So it's really quick invasion. Egress takes more time to set up because the parasite has to finish its cell division, and the way it divides its cell is very different from the way we divide our cells. So our division usually is binary, mitosis. It goes 1 to 2. Parasite goes from 1 to 24. And this process is called schizogony. And what the parasite does, during this process, it starts sending enzymes that are called proteases into the vacuole that it stays within the red blood cell. So one thing I sort of didn't mention is when it goes into the red blood cell, it wraps the red blood cell membrane around itself in a protective cocoon called the parasitophorous vacuole. And so to get out now, it has to break the vacuole and then the red blood cell membrane, both things, right? And so what it does is, while it's making its daughter cells, it starts sending out these little packets of proteases. And they're sent by these vesicles. What triggers that pathway is still a little unclear. We don't know how the parasite knows, okay, it's time for me to start preparing to get out, and I'll start sending these vesicles out. And what we have done, and this is basically unpublished, but I'm more than happy to talk about it because we've talked about it a lot at conferences and so on. which is that the parasite, we've started looking at this process using fluorescence microscopy. And we're looking at live parasites. So we can see how they're sending these little packets out into the host, into the vacuolar space. And then at some point it decides, okay, time for me to break out of it. And what happens is very interesting. The vacuolar membrane, which is usually pretty irregular in shape, suddenly becomes very spherical. So it adopts like really a circular shape. And 30 seconds after that circular shape is adopted, we see one single point where the vacuolar membrane breaks, releasing the parasites into the red blood cell. What triggers it is a huge mystery. The field has been talking about it and trying to figure out, not just us, several groups around the world have been trying very hard to figure out how the parasite knows it's time to leave. And our data suggests that it's actually not just once that it decides to leave. It has two checkpoints where it seems to say, okay, now I'm going to start sending on these proteases. And several hours later, it says, now it's time to break the vacuolar membrane. And that timing and those triggers are something we and others are trying to figure out how the parasite does that.
Well, I think there's only one thing left, and we alluded to it in the intro, and that was the story of a parasite messing with the wrong college students. So perhaps you could enlighten us as to that story.
Oh, yeah. This is a crazy story, but it's true. One day, a young college student shows up, gets up in the morning, has this little bit of fever, sort of flu-like symptoms, decides, I can't miss college today. I have this really important lab module that I need to be there. So just pops a couple of Tylenol, goes into lab. What are we doing in lab today? Well, we're going to do blood smears and do different types of stainings to identify all the little cells that are in your bloodstream. And the way we did it was the college student pricked his finger and smeared a bunch of slides with the little drop of blood on his finger. And then we stained with a bunch of different things, and then each of us had to look under a microscope and identify blood cells. I was good at it. I identified all the monocytes and the neutrophils and everything. And then I noticed something funny. In one of my stains, I noticed my red blood cells, a couple of them, seem to have a nuclei. And obviously, if you know anything about red blood cells, you know they have no organelles. It's an empty bag full of hemoglobin. Well, not my red blood cells, apparently. And so I called my professor, my same professor I'd mentioned earlier, Professor Nair. She came over and I said, Dr. Nair, I think my blood cells have nuclei. And she just gave me this very disappointed look and saying, Vasant, red blood cells don't have nuclei. Just move over. Let me take a look at your slides. You're probably just misidentifying something. And she looks at it, and then she comes over and she says, oh no, you need to go to a hospital right now.
You had a squatter in your red blood cell, didn't you?
I did. I did not know that. And so she rushed me off to a hospital. And sure enough, before I got the results that evening, I got what's a classic symptom of malaria, chills. So you get this uncontrollable shivering, about 5 minutes or so, where your whole body shivers. And why does it shiver? Now I know. I didn't know at that time. That's egress. So your red blood cells are bursting, releasing all these baby parasites back into your bloodstream to infect new red blood cells. And when you lose that many blood cells, your body's response is shivering. So as soon as I got those shivers, nothing else, no other infectious agent on the planet can cause that except malaria. As soon as I got that, before the hospital called and said I had malaria, I knew I had malaria. I was lucky. I had good access to medical care, and so I got treated. I was lucky in more than one way. I had Vivax and falciparum at the same time. This does happen in the field.
Always an overachiever.
Of course, if you're going to get malaria, might as well get both at the same time and never get it again, right? And so I've been lucky. So yeah, I wish I had saved those parasites. Now, if I'd go back and collect my own parasites, I would do that. And I have used my own blood to grow parasites in the lab. So I know my blood can sustain parasites, both in the field and in the lab.
Well, parasites out there, let that be a lesson to you. And our guest today has been Dr. Vasant Muralidharan, Associate Professor in the Department of Cellular Biology in the Center for Tropical and Emerging Global Diseases. Vasant, thanks for being with us.
Thanks, Kim. Thanks, David, for inviting me. This was a real pleasure.
So links to additional information about Dr. Muralidharan's research can be found on our Instagram account, which is at PPPPodcastUGA.
As always, we appreciate you tuning in today. And if this is your first time listening to the People, Parasites, and Plagues podcast, we encourage you to check out the other exciting episodes launched this season. Please rate the podcast, and if you have any questions or suggestions for future episodes, reach out to us via email at ppp@uga.edu. This podcast is brought to you by the Faculty of Infectious Diseases and the Grady College of Journalism at the University of Georgia. It is supported by the University of Georgia through the Office of Research, the UGA Graduate School, and the College of Veterinary Medicine. Thanks to the New Media Institute at Grady for use of their Studio Not Found podcast facilities, and a special thanks to our production assistant, Sid Wigand, for research, editing, scheduling, and keeping us on track, as always.
That's a wrap.